Good day, everyone. Welcome back to the third lecture of the biology curriculum entitled Energy. I am Aaron Ezekiel Heramia, and I am one of the instructors of the Department of Biology of CERTS. So, in this part, we will talk about the metabolic pathways. These are the biological processes that produces and utilizes energy. So, to be more specific, we will talk about these two main topics. So photosynthesis and cellular respiration. So before we talk about the metabolic pathways mentioned a while ago, let us talk about first the keywords that we need to understand before going deeper to these metabolic pathways. So let us define these terms. First is metabolism. Metabolism is defined as the sum of chemical processes in an organism. So recall your first topic or your first lecture, which is the characteristics of life. Part of part of it is metabolism. So all of the chemical processes happening in your body is a part of metabolism. Metabolism is the summation of all of these processes that occur inside of you. So digestion, respiration, excretion, and etc. These are part of metabolism. Next is metabolic pathway. It is a series of chemical reactions in a cell that build or break down molecules for cellular processes. So these metabolic pathways uh, are involved in building up or breaking down molecules necessary for cellular processes. So we have two types of metabolic pathways. We have anabolism and catabolism. So anabolism is the building up of molecules. Catabolism is breaking down of molecules. So when we build up our molecules, when we build up uh, compounds, it is in it is an anabolic pathway or anabolism. So when we break down these molecules, we break down some compounds. This is an example, or this is a catabolic pathway or catabolism. So organisms like us humans and other species can be divided based on the type of based on the procedure on how he acquire energy or acquire energy. Again, an organism can be classified based on how she, or it acquires energy. So we have two, autotrophs and heterotrophs. Autotrophs are organisms that produce its own food. So they manufacture their own food using whether inorganic or organic compounds. So under autotrophs, we have two main types, chemoautotroph and photoautotroph. Chemoautotroph uses chemicals to produce food, so whether organic compounds or inorganic compounds. Photoautotroph uses radiant energy to produce own food. So photoautotrophs usually utilizes the energy from the sun or the sunlight to produce their own food. So we will talk about that later on with photosynthesis. Now, heterotrophs are organisms that can produce its own food. So they depend on other uh, sources. So they cannot manufacture their own food, just like unlike autotrophs. There are many types of heterotrophs. For example, carnivores depends on meat of other organisms. Omnivores are depends on both plant and animal sources, and herbivores that depends on plant sources only. So there are many types of heterotrophs. Those are only the examples of it. Lastly, we have two types of reactions that and there are two types of reactions that we need to understand for, for us to fully there are two types of reactions that we need to define in order for us to understand the processes that we will gonna talk about later on. So endergonic reaction and exergonic reaction. So endergonic reaction are chemical reaction is a chemical reaction that require energy. Meanwhile, exergonic reaction is a chemical reaction releasing energy. So ender requires and exer releases. Now we will talk about adenosine triphosphate. So ATP is the energy currency of the cell. It stores and releases energy required for cellular process. So the structure of the adenosine triphosphate consists of adenine, five carbon sugar named ribose, and three phosphate groups. So why is ATP very relevant in our topic for this presentation? So ATP acts like a battery. So in the processes that we will talk about later, it is continuously broken down from ATP and ADP, which is adenosine diphosphate, to adenosine diphosphate two has two phosphate groups instead of three, and ADP is then regenerated to ATP. So it acts like a battery because ADP is like a partially charged battery. And then when it is regenerated to ATP, it becomes fully charged. So phosphorylation is the transfer of third phosphate group to a substrate in order to release energy. So again, uh, phosphorylation is the transfer of the phosphate group. So why is phosphorylation uh, connected in or relevant in this study? So phosphorylation we will talk about later because phosphate group is always transferred in order for us to release or produce energy. So we will understand it later on why phosphorylation is very crucial. ATP is associated with exergonic glucose catabolism and the endergonic pathways that power living cell. So where do we acquire our energy in the first place? As humans, for example. So we get our um, energy from the food we eat. And that is why ATP is very associated with glucose catabolism. Because ATP is again the energy currency of the cell. And glucose catabolism is an important process for us organisms, especially us um, aerobic organisms, which we'll talk about later on. And the endergonic pathways that produces energy also requires the use of ATP. Although ATP is a great molecule for transferring energy. It is not a good compound, or it's not good one for storing large amount of energy. That is why we need glucose, because glucose stores more than 90 times of the chemical energy. Or rather, a single molecule of sugar glucose or glucose store more than 90 times the chemical energy of a molecule of ATP. So it is more efficient for cells to keep only a small supply of ATP on hand. So now we will move on to the metabolic pathways that we have. First, we will talk about photosynthesis. So photosynthesis, when we break down this term, we have two two parts. Yes, photo and synthesis. So photo means light, synthesis meaning production. So from the word itself, it means the production using light. So production using a light source. So photosynthesis is the only biological process that converts external energy from outer space, which is sunlight, to chemical compounds. Photosynthesis is very essential to all life on Earth, not only to autotrophs. Why? It is because organisms depend on plants, not only because it is a food source, but also plants produce oxygen for our cellular respiration, and also, yeah, photosynthesis produces sugars and carbohydrates for our uh, energy, for energy production, for energy consumption. That is why photosynthesis is very crucial for all organisms, not only for autotrophs. So it requires sunlight, carbon dioxide, and water as substrates to produce glyceraldehyde 3-phosphate for energy, and oxygen as waste product. So the main product for this reaction is glyceraldehyde 3-phosphate, which is a simple sugar. And then this simple sugar will then be converted to more complex sugars such as sucrose, fructose, maltose, and so on. So just recall your biochemistry or biochemical compounds. Yeah. So for photosynthesis, we have our equation, which is 6 molecules of carbon dioxide plus 6 molecules of water when ayan, again 6 molecules of carbon dioxide plus 6 molecules of water when we combine this with the energy coming from the sunlight, we will obtain a molecule of glucose, which is 6 C6H12O6 plus 6 molecules of oxygen. So that is the overall equation for the photosynthesis reaction. So the sites of photosynthesis. So where do photosynthesis occur? So in plants, it is happening or occurring in the mesophyll. Mesophyll is the middle layer of the leaf. And on the mesophyll, it contains stoma, which is the openings where gas exchange occurs and which regulates water balance. So stoma are tiny openings along or inside. It's not inside, on the mesophyll. These are tiny openings where gas exchange. exchanging of carbon dioxide and oxygen occur. So stoma are regulated by guard cells by swelling and shrinking. So it regulates the opening and closing of the stoma. So in eukaryotic autotrophs, such as plants and other organisms, we have this organelle called chloroplast. This is the main site of photosynthesis. So inside the chloroplast, we have thylakoids. What are thylakoids? Thylakoids are disc-shaped structures containing thylakoid lumen. So thylakoids are arranged in a stack called granum, or plural is grana. And surrounding grana inside the chloroplast, we have a liquid filled space called stroma. So photosystems are light collecting units of the chloroplast. These are proteins in the thylakoid membrane organizing chlorophyll and other pigments into clusters. So, mentioning light and pigments, let us focus now on the light and pigments. So, as we have talked in the first part of this presentation or this lecture, the sunlight, our sun, our soul, the the closest star of the Earth emits electromagnetic waves, which exhibits radiant energy. And these electromagnetic waves can appear in many different, or it can appear in different ways. So one particular one particular electromagnetic wave that we will discuss is visible light. It is a fraction of the electromagnetic spectrum that can be perceived by the human eye. So, yeah, the electromagnetic spectrum is a spectrum of electromagnetic waves based on the wavelength of based on the wavelength of these electromagnetic waves. So visible light is a fraction of the electromagnetic spectrum which we can perceive by our naked eyes. So this visible light is very important in photosynthesis as it this white light or this visible white light is where plants acquire their radiant energy for energy production. Now, the pigments that we will talk about for this photosynthesis, we have two, chlorophyll and carotenoids. Chlorophyll is a green pigment found in chloroplast. So we have four main types of chlorophyll, but we will only focus on the two, chlorophyll A and chlorophyll B. So chlorophyll A absorbs red wavelengths of the visible light, and chlorophyll B absorbs blue-violet wavelengths of the visible light. So this absorption of red and blue-violet wavelengths causes the reflection of green and yellow wavelengths. That is why green appears, I sorry, leaves appear green because green is reflected and not absorbed by this chlorophyll pigment. Light energy is absorbed into electrons, which raises its energy level. So the light energy coming from the sun causes the electrons of chlorophyll to highly energize or to be excitable. Aside from chlorophyll, we have carotenoids, which absorbs excess energy from light-dependent reactions. So sometimes when chlorophyll absorbs too much light, electrons gets too excited and may produce excess energy. So these carotenoids absorbs the excess energy coming from the light absorption of chlorophyll and the excess energy coming from light dependent reactions that we will talk about in the next slide. Next slides. Now we will focus or we will talk about electron carriers. So when chlorophyll absorbs sunlight, its electrons gain deal amounts of energy requiring electron carriers. So imagine you have a hot coal in your backyard. So you as a person will not pick that hot coal because first of all it is very hot, and by that you will require some instrument to pick up that hot coal. So that is what electron carriers job is. It is used to uh, receive or accept highly energized electrons coming from the light absorption of chlorophyll. So carrier molecule accepts high energy electrons to be transferred elsewhere in the chlorophyll in a process called electron transport. So a carrier molecule, as the definition mentions, accepts high energy electrons. And the chain of carrier molecule or the chain of carrier molecules in a membrane is called electron transport chain. So they transport electron elsewhere in the chlorophyll for future processes. So an example of a carrier molecule or of an example of an electron carrier is called an ADP or nicotinamide adenine dinucleotide phosphate. It carries two electrons and a hydrogen ion, converting into NADPH. So this NADPH is very important because it carries high energy electrons produced by light absorption in chlorophyll to chemical reactions elsewhere in the cell. So these high energy electrons are used to help build a variety of molecules the cell needs, including carbohydrates like glucose. So now we will talk about the reactions that happening in photosynthesis. So we have two, light-dependent reactions and light-independent reactions. So from the term itself, light-dependent reactions requires water and requires sunlight, requires the presence of sunlight. Aside from sunlight, it requires water or H2O. So this light dependent reaction occur at or this dependent reaction occurs at the thylakoids or grana. Light dependent reaction produces ATP, NADPH, and oxygen. So let's specify the steps in light dependent reaction. So first, light and light absorbed by photosystem 2 is used to break up water molecules into energized electrons, hydrogen ions, and oxygen. So photosynthesis begins when pigments in photosystem 2 absorb light. So why is photosystem 2 uh, why is photosystem 2 named photosystem 2, and why is photosystem 1 named as photosystem 1? Because photosystem 1 is first described before. I Photosystem 1 is first discovered, and after that it was photosystem 2 that was discovered. So it is just a matter of discovery, the time of discovery of this photosystem. But photosystem 2 will first photosystem 2 uh, photosys I mean photosynthesis starts at photosystem 2. The light energy is absorbed by electrons, increasing their energy levels. These high energy electrons are passed onto the electron transport chain, or the chain of carrier molecules. So does the chlorophyll run out of electrons as light continues to shine? No, it does not. The thylakoid membrane contains a system that provides new electrons to chlorophyll to replace the one it has lost. So these new electrons come from the water molecules that we are requiring for this reaction. So enzymes on the inner surface of the thylakoid membrane break up each water molecule into two electrons, two hydrogen ions, and one oxygen atoms. So the two electrons replace the high energy electrons that chlorophyll has lost to the electron transport chain. As plants remove electrons from water, oxygen is left behind and released into the air. So that is where oxygen is being released as a product. This reaction is the source of nearly all the oxygen in Earth's atmosphere, and it is another way in which photosynthesis makes our lives possible. The hydrogen ions left behind when water is broken apart are released inside the thylakoid membrane. So far we have released oxygen atom, which is very crucial for organisms such as humans and other animals. So that is why we are also benefiting on this photosynthesis. So the first step, we'll just remember that photosystem 2 absorbs light and it will break up water molecules into energized electrons, hydrogen ions, and oxygen, which oxygen will be released into the atmosphere as a byproduct. Next, high energy electrons from photosystem 2 move from the electron transport chain to photosystem 1. So after we have broke down the water molecules into electrons, hydrogen ions, and oxygen, the highly energized electrons will move from photosystem 2 to photosystem 1 through the electron transport chain. So the energy from the electrons is used by the molecules in the electron transport chain to transport hydrogen ions from the stroma into the inner thylakoid space. So after that, electrons released by the photosystem 2 are energized again in photosystem 1. Enzymes in the membrane use the electrons to form NADPH. NADPH is used to make sugar in the Calvin cycle. So the pigments in photosystem 1 use energy from light to re-energize the electrons. So not P then picks up these high energy electrons, along with hydrogen ions that we have produced in the second step, on the outer surface of the thylakoid membrane, plus on each ion that becomes NADPH. So next, the inside of the thylakoid membrane fills up with the positively charged hydrogen ions. This action makes the outside of the thylakoid membrane negatively charged and the inside positively charged. So as we are re-energizing the electrons that is transported from photosystem 2 to photosystem 1, uh, hydrogen ion or hydrogen ions passes through the thylakoid membrane. That is why it becomes more positive. Uh, it becomes more positive inside the thylakoid space and negative. I mean, inside more positively charged and outside negatively charged. And the difference in charges across the membrane provides the energy to make ATP. That is why the hydrogen ions are so important. So it is very important to have our electrochemical gradient in order for us to produce ATP. So for the last step in our light-dependent reaction, as hydrogen ions pass through the ATP synthase, their energy is used to convert ADP into ATP. So what is ATP synthase? So ATP synthase is a protein that allows hydrogen ions to pass through it. And then, as hydrogen ions pass through the ATP synthase, it converts ADP into ATP. How do it converts ATP? How do it converts ATP into ATP into ATP? So ATP will receive a phosphate group coming from uh, yeah, ADP will receive a phosphate group together to produce ATP as it allows hydrogen ion to pass through. So because of this system, light-dependent electron transport produces not only high-energy electrons but also ATP as well. So in summary, to summarize this reaction, the light-dependent reaction use water, ATP, and NADP to produce oxygen and two high-energy compounds, ATP and NADPH. So ATP and NADPH is very crucial for our next step because without ATP and NADPH, uh, the Calvin cycle will never occur. And that is why light-dependent reaction is also vital for our next step. It has an important role to play in the cell. It will provide the energy to build energy-containing sugars from low-energy compounds. Now we will move on to light-independent reactions or Calvin cycle. So Calvin cycle is named after the American scientist Melvin Calvin, who worked out on the details of this remarkable cycle. So the Calvin cycle occurs at stroma, or the liquid filled space inside the chloroplast. It requires NADPH, ATP, and carbon dioxide. It produces sugars for our energy, so energy production. So now we will talk or we will see how the Calvin cycle works. So first, carbon dioxide enters the cell. Six carbon dioxide molecules are combined with six molecules of ribulose biphosphate, resulting to 12 molecules of 3-PGA. 12 molecules of 3-PGA or phosphoglyceric acid. This carbon fixation is catalyzed by rubisco or ribulose biphosphate carboxylase. Okay, so this process is called carbon fixation because it is a process of fixing carbon from an inorganic source into an organic source. So our inorganic source for this process is carbon dioxide, and we are fixing it or we are fixing the carbon into an organic source, which is the 12 molecules of 3 phosphoglyceric acid. So yes, the 6 carbon dioxide will be combined with RuBP to produce the molecules of 3-PGA, that is carbon fixation. Next, energy input. Energy from ATP and high energy electrons from NADPH, sorry for the correction, that is NADPH, are used to convert from NADPH. I'm sorry. There's a corrigendum on our PowerPoint. So energy from ATP and high energy electrons from NADPH are used to convert the 12 3 carbon molecules into high energy form. So the 3-PGA that we have produced in carbon fixation uh, is used to, I mean, the 3-PGA that we have created in the carbon fixation will be converted into high energy form by the high energy electrons that is accepted or acquired by NADPH and also by the energy from ATP. So next, 6 carbon sugar produced. So after that, 2 GA3P or glyceraldehyde 3-phosphate molecules are removed from the cycle to produce organic compounds such as sugars, lipids, and amino acids. So after we have produced the 3-PGA molecules and converting it into higher forms, we are now uh, we are now yielding 6 carbon sugar. So this the two of it will be removed to produce organic compounds such as sugars, lipids, and amino acids. Now, what about the 10? As we have produced 12 molecules of the three, the 12 GA3P molecules, we have produced 12. So what happens to the 10? So the 10 remaining GA3P molecules are converted back into 6-RuBP for the next Calvin cycles. So this regeneration of the 5 carbon molecules is very essential because Calvin cycle does not occur only once because it is a cycle. It is never ending. It will happen again and again as long as the plant survives. That is why we need the majority of the GATP for the next Calvin cycles to that will happen in the future reactions. So as we can see, the Calvin cycle uses six molecules of carbon dioxide, which is an organic compound, to produce a single six-carbon sugar molecule. As photosynthesis proceeds, the Calvin cycle works steadily, removing carbon dioxide from the atmosphere and turning out energy-rich sugars. So this energy-rich sugars is needed to meet its energy needs and to build more complex macromolecules such as cellulose that it needs for growth and development. So when us organisms or when other organisms like us humans and animals eat plants or consume plants, they can also use the energy stored in the carbohydrates produced in this cycle. So just the summary of the photosynthesis. We have two light-dependent reactions and Calvin cycle. Light-dependent reaction is the process that converts light into water, ADP, and NADPH. So the light energy coming from the sun undergoes through the electron transport chain, which releases oxygen and produces ATP and NADPH that is essential for the next step, which is the Calvin cycle. So the Calvin cycle is the process of fixing the carbon dioxide, which is an organic compound, into organic compounds for energy. So the Calvin cycle has three steps: carbon fixation, which is combining of carbon dioxide molecules with five carbon molecules or RUBP to produce 12, um, 12 three carbon molecules or PGA. So this 3 PGA will then be converted into Ga3P through the use of the energy from ATP produced in light dependent reaction and the electrons, high energy electrons from NADPH. So as we have produced 12 molecules of Ga3P or glyceraldehyde 3 phosphate, two of the molecules will then be removed from the cycle to produce sugars, lipids, and amino acids for energy, while the 10 of the remaining Ga3P will be converted back into RUBP for the next or for the future Calvin cycles to occur. So that is the summary of the photosynthesis. So it is very simple to understand, and we will not delve too much on the process. So you just need to understand that light-dependent reaction requires light and H2O, and Calvin cycle requires the products in light-dependent reaction along with carbon dioxide. Next is cellular respiration. Cellular respiration is the reverse of photosynthesis. Why? So we will understand later why it is called as reverse of photosynthesis. So cellular respiration requires glucose and oxygen for breakdown to produce energy and other products such as water and carbon dioxide. So before in photosynthesis, we are producing, we are building up molecules or we are building up compounds. It is a anabolic pathway to produce energy. Now, in cellular respiration, we are breaking down these compounds, these molecules, to produce energy. That is why it is called as reverse of photosynthesis because it breaks down the products that is produced in photosynthesis to produce energy. So, cellular respiration has products such as water and carbon dioxide. So, typically, these are waste products. So, glucose is derived from the food we eat, mainly from plants. So, yeah, as we have talked a while ago, plants are the main source of glucose because of the photosynthesis, because they are producing glucose in this process, in that process, which is photosynthesis. That is why in cellular respiration, glucose is now being broke down to produce the energy we need. That is why cellular respiration and photosynthesis highly codepend with each other. When we produce glucose in photosynthesis, we are building up the glucose in photosynthesis. We will now break down the glucose that is produced in cellular respiration. So cellular respiration has two types, aerobic and anaerobic. So what does these two words mean? Aerobic reaction means that oxygen is present, while anaerobic reaction or respiration means that oxygen is absent or it is not required in the process. So aerobic respiration involves glycolysis, Krebs cycle or citric acid cycle, and electron transport chain. So the overall equation for this cellular respiration is C6H12O6 plus 6 molecules of oxygen produces 6 molecules of carbon dioxide and 6 molecules of water. So, as you can see, it is really the reverse of photosynthesis because in photosynthesis we are producing glucose and oxygen from carbon dioxide and water. Now we are producing carbon dioxide and water from glucose and oxygen. So now we will talk about the major steps in cellular respiration. So first is glycolysis. It occurs at cytosol of a cell. It requires glucose, 2 ATP, and 2 NAD. NAD meaning NAD means nine na nicotinamide adenine dinucleotide. So NAD is again an energy or electron carrier. So it produces two pyruvate, two net ATP, and two NADH. Net ATP means that we are reduced the ATP that we have used, but originally it produces 4 ATP. So 4 ATP minus the 2 ATP that glycolysis requires is equal to 2 net ATP. So glycolysis is the first set of reaction of cellular respiration. Glycolysis is the process in which one molecule of glucose is broken in half, producing two molecules of pyruvic acid or pyruvate, which is a three carbon compound. So glycolysis doesn't require oxygen. That is why it can occur in both aerobic and anaerobic organisms. So glycolysis can be divided into two stages: energy requiring stages and energy releasing stages. So energy requiring stages requires two ATP molecules to split glucose into three carbon molecules. And energy releasing stage, in which NAD or NAD receives high energy electrons yielding to two NADH. So that GA3P that we have produced in energy requiring stage is then converted into pyruvate, and ATP is not required but produced along this stage. So let's use a diagram to further understand glycolysis. So first we have glucose. So glucose now, the ATP that is required in the glycolysis, as we have mentioned, the phosphate group in this ATP will then be uh, or it will separate the one phosphate group from each ATP that is required will separate from the main ATP, which is be converted into ATP, and this phosphate group that was being separated will um, combine with our glucose. So we have two phosphate groups combined with the glucose, and this will form fructose 1,6 biphosphate, which is an unstable form of our uh, glucose. So because it is unstable, we need it to further separate or split it into two. So splitting of fructose 1,6 biphosphate will produce two types of three carbon compounds, glyceraldehyde 3-phosphate and DHAP. So DHAP is not needed in the process of cellular respiration. It is undesirable. So GA3P is the one that we needed in cellular respiration, and it will continue to the next step. But what happens to DHAP? So we will not worry with DHA3P because it is easily converted into GA3P, which is desirable three carbon compound for cellular respiration. So now we will have two glyceraldehyde 3-phosphate into the next step. So this the previous stage was uh, energy requiring because we needed ATP to occur for the process to occur. We need ATP for the stage to occur. Next is the uh, energy releasing stage because now we will release ATP along this stage. So as we have mentioned, the high energy or highly energized electrons will be combined with NAD or NAD to produce NADH. And then along the way, ATP will be produced from ADP or adenosine diphosphate to ATP or adenosine triphosphate. So after that, pyruvate or three carbon molecule will be produced. So this process or energy releasing stage will happen twice. That is why we will it will be producing four ATPs and two NADH. So removing the two ATPs that we have required, we will only arrive into two ATP, two NADH, and two molecules of pyruvate. So next, we have Krebs cycle. Krebs cycle occurs at mitochondria. It requires the two pyruvate, two NAD, and two coenzyme A. It produces 6 carbon dioxide, 8 NADH, 2 FADH, and 2 ATP. So at Krebs cycle, we have two main processes or two main general stages. It is the citric acid production or pyruvate oxidation that we can call. It is the oxidation of pyruvate to transform or convert into citric acid. And the citric acid cycle itself. So citric acid production, what does this stage means? So let us use a diagram again to to fully understand the the pyruvate oxidation. So pyruvate, as we have produced in glycolysis, will then uh, pyruvate will then what happens to pyruvate? Uh, yes, okay, sorry. Pyruvate will enter the mitochondria because it was produced in the cytoplasm of the cell. And one carbon of the pyruvate will be removed, and this separated carbon will produce carbon dioxide. So coenzyme A will then join the two carbon molecule forming coenzyme or acetyl CoA. So the coenzyme will combine will be combined with the two carbon molecule to form acetyl CoA. So this acetyl CoA then will be combined to for carbon compound forming citric acid. So again, from pyruvate will separate a carbon molecule forming, and this carbon molecule separated will form carbon dioxide to be released, and this will leave us with two carbon molecule. So this two carbon molecule will combine with an enzyme called coenzyme A, and this enzyme substrate will be called as acetyl-CoA. This acetyl-CoA will then add into a four carbon compound forming citric acid. After that, citric acid will undergo into citric acid cycle. As the cycle continues, citric acid is broken down into a four carbon molecule. More carbon dioxide is released and electrons are transferred to energy carriers. So first, let us look with the six uh, carbon atoms in the citric acid. So one will be removed uh, primarily to produce carbon dioxide. Then we will leave us with five carbon molecules. Again, another molecule of another carbon will then separates the compound to form carbon dioxide, leaving us with four carbon molecule. So this four carbon molecule is ready to accept again another two carbon acetyl group, which starts the cycle all over again. And now what happens to the electron carriers that is required also in Krebs cycle? So the NAD that is required will continuously accept highly energized electrons along the way, forming NADH. So we have in pyruvate oxidation, we have produced one NADH, and in citric acid cycle, we have produced one, two, three, three NADH. So they are accepting highly energetic electrons along the way. Also added to that, we have another energy or electron carrier named as FAD or flavine adenine dinucleotide. So flavine adenine dinucleotide also accepts high energy electrons, forming FADH or flavine adenine dinucleotide hydrogen. So this energy our electron carriers will be very essential for our last step because these electron carriers are required in production of ATP, which is vital for energy. So when we exhale, we are exhaling our carbon dioxide. So this carbon dioxide is the product of this Krebs cycle. That is why it is called cellular respiration because we are using oxygen and glucose in the previous uh, steps of this cellular respiration, and we are expelling the products or the waste byproducts, which is carbon dioxide, as we go on to this process. That is why it is called cellular respiration. So lastly, we have electron transport chain. It occurs at mitochondria. It requires NADH and FADH and produces ATP and water. So this electron transport chain uh, produces majority of the ATP in the cellular respiration. So how do electron transport chain work? Uh, Recall the light dependent reaction because electron transport chain is very similar. No, it's very similar with light dependent reaction. So first, electron transport at carrier proteins. So high energy electrons carried by NADH and FADH2 passes through proteins. At the end, oxygen serves as the final electron acceptor, forming water. So the NADH and FADH that we have produced will travel through carrier proteins in the inner membrane of the mitochondria. So the NADH will then be converted into NAD and H, and FADH2 will be converted into fat. So what happens to the separated hydrogen electrons? Hydrogen electrons will then be pumped inside the intermembrane space by these proteins in the inner membrane. So as we are separating it from the electron carriers, H or the electrons or H or hydrogen ions will be then pumped. So as travels through this electron transport chain, hydrogen ions are continuously pumped inside the intermembrane space. So every time two electrons travel through ETC, their energy is used to transport hydrogen across the membrane, making the intermembrane space positively charged. So as I have mentioned, when electrons are traveling into the electron transport chain, which is carried by NADH and FADH2, hydrogen ions, also carried by the NADH, will then be pumped into the intermembrane space. So the accumulation of hydrogen ions causes the intermembrane space to become positively charged, and the outside, which is the mitochondrial matrix, becomes negatively charged. And ATP production. So, how does ATP produce in this step? So, hydrogen accumulated at the intermembrane space escapes through channels called ATP synthase, which converts ADP into ATP. So, when hydrogen is now accumulating inside the intermembrane space, the hydrogen will now escape through a protein called the ATP synthase. So let's go back to the ATP synthase. So this is the ATP synthase. ATP synthase allows hydrogen ions to pass through. And when hydrogen ions passes through this ATP synthase, it produces ATP by combining a phosphate group into ADP. That is why majority of the ATP is produced in this step. So to summarize the cellular respiration, first we have glycolysis. Glycolysis is the breakdown of glucose into pyruvate, and it also produces electrons, it produces NADH from the highly energized electrons that will be needed in oxidative phosphorylation, which is electron transport and chemiosmosis. So that is glycolysis, the breakdown of glucose into pyruvate. So this glycolysis will produce 2 ATP. Then the pyruvate from glycolysis will proceed to pyruvate oxidation. This pyruvate oxidation will remove one carbon from the three carbon molecule, which the separated one carbon molecule will be formed as carbon dioxide, which is the waste product of cellular respiration, and the remaining two carbon molecule will be combined with with an enzyme called coenzyme A, producing acetyl coenzyme. Acetyl CoA. So yeah, acetyl CoA. So acetyl CoA will be combined with the four carbon molecule four carbon compound, which will be which will then produce citric acid. So citric acid will go on to citric acid cycle. Citric acid cycle is the continuous cycle of removing carbon molecules in citric acid to produce CO2, and also the receiving of highly energy electrons of electron carriers such as NAD and FAD to produce NADH and FADH, which is again essential or required for our next step, which is electron transport chain or oxidative phosphorylation. So the citric acid cycle, we only need to remember that from citric acid we will remove a carbon molecule to produce five carbon compound, and then from the five carbon compound we will gonna remove again a carbon molecule to produce a four carbon compound. So the final form of this Krebs cycle is ready to be combined again with uh, acetyl CoA, which is for the next process. So it is a cycle. It is a cycle. 654, and then this 4 carbon compound will be readily combined with acetyl CoA for the next Krebs cycle. So in this Krebs cycle, or the overall Krebs cycle, will yield us 2 ATP. Lastly, oxidative phosphorylation, which is electron transport and chemoosmosis, is the production of the majority of the ATP in cellular respiration. It involves the transport of electrons coming from glycolysis and citric acid cycle and production of ATP due to the hydrogen ion accumulation from electron transport. So with this step, we will yield, or we are yielding 34 ATP. So some books, they they write ETC as 32 ATP, or they say that ETC produced 32 ATPs at the end. So it is not set in stone. It can be 32, 33, or 34. So at the end, 34 plus 32 plus 32 will yield us into 38 ATP, or in other books it is 36, because yeah, some of them write 32 in oxidative phosphorylation. So that is the whole summary of cellular respiration. I hope you understand the whole process of cellular respiration, though it is very simple. It is the simple uh, version of cellular respiration. We just need to understand that from glucose, it will be converted into pyruvate, acetyl CoA, citric acid, and then this process will produce or this process will then remove highly energy electrons that are carried by electron carriers NAD and FAD to produce NADH and FADH. So this NADH and FADH will then carry high energy electrons to the electron transport chain to undergo electron transport. An electron transport will then allow us to produce ATP through the accumulation of hydrogen ions inside the intermembrane space. So yeah, that is the whole process of cellular respiration. So as we have mentioned, there are two types of organisms and there are two types of respiration that under uh, go along with them. So aerobic respiration goes with aerobic organisms, and anaerobic respiration goes with anaerobic organism. So we have talked about the cellular respirations going on aerobic organisms. Now let's talk about the anaerobic organisms. So what happens when we don't have oxygen? When an organism does not need oxygen. So how does it undergo cellular respiration? So anaerobic organisms can also undergo glycolysis because glycolysis does not require oxygen, as we have mentioned. But after glycolysis, where do they what processes do they undergo after? So they undergo fermentation. So fermentation occurs in anaerobic organisms. So they this process releases energy by producing ATP without the use of oxygen. So after glycolysis, uh, anaerobic organisms, usually prokaryotic organisms, undergo fermentation to produce ATP without the use of oxygen. So we have two types of fermentation. We have alcoholic fermentation and lactic acid fermentation. So alcoholic fermentation, in its own term, from the term itself, it produces alcohol in the form of ethanol and carbon dioxide. So it is present in microorganisms such as yeast. A leavening agent causing bread dough to rise. So when when you observe a bread, when we cut it in half and we observe it inside, there are many tiny pockets of air, tiny pockets that we can observe, can see. So these tiny pockets is the manifestation of alcoholic fermentation when we use yeast as a leavening agent. So when yeast or Saccharomyces cerevisiae consumes glucose from the cooking process of bread, when it consumes sugar, it will then produce carbon dioxide in in order for the dough to rise. So when yeast in the dough runs out of oxygen, it begins to ferment, giving off bubbles of carbon dioxide that form the air spaces you see or we see in the size of bread. So along with that, a small amount of alcohol produced in this kind of fermentation will evaporate when the bread is baked. So that is why we are not being drunk with bread, because only small amount of alcohol is produced in fermentation or alcoholic fermentation when baking bread. So next type of fermentation is lactic acid fermentation. So now in this type of fermentation, what uh, differs from alcoholic fermentation, it converts pyruvate into lactic acid. In alcoholic fermentation, we have converted pyruvate into alcohol in the form of ethanol. Lactic acid fermentation converts pyruvate to lactic acid. So it is called lactic acid fermentation because it produces, you guess it, lactic acid. So lactic acid is produced in your muscles during rapid exercise when the body cannot supply enough energy and when the body cannot supply enough oxygen to the tissues. So when we exercise, when we do rigorous activities, the blood cells of the skeletal muscles may contain insufficient supply of oxygen. That is why it resorts to lactic acid fermentation. The buildup of lactic acid causes a painful burning sensation of us, causing stiffness in our muscle cells. And yeah, so that is why we are getting sore after doing some exercise, sports, and rigorous activities. So lactic acid fermentation is also present in prokaryotes that are used to make dairy products such as yogurt and cheeses. So this is lactic acid fermentation. So that is all for our lecture. Thank you so much for listening, and I hope you've understand this lesson very well. Until next time, this is Ir Ezekiel Haramia, and thank you very much for listening.